Good evening, ladies and gentlemen. I've been asked to introduce myself before introducing the speaker, which is the main purpose of my being here. I'm Jean Thomas. I'm the new biological secretary of the Royal Society, and I'm Professor of Biochemistry in Cambridge. The other thing I've been asked to do, before I have the pleasure of chairing this lecture, is to ask you all to switch your mobiles off, if you would, please. And also to tell you that this lecture will be recorded and webcast. So this is the Francis Crick Prize Lecture we're about to hear. This was established in 2003 through an endowment by Dr. Sidney Brenner, Commander of Honour and Fellow of the Royal Society, who was the joint winner of the Nobel Prize for Physiology or Medicine in 2002. The lecture is delivered annually in any field of biological sciences, but preference is given to the general areas in which Francis Crick himself worked, genetics, molecular biology, and neurobiology. And you'll see that this evening's speaker is eminently well suited to talk to us. So the Francis Crick lecturer for this year is Dr. Simon Fisher. Dr. Fisher is a Royal Society research fellow, so he's a fellow of one of our flagship schemes for young scientists in the Society, and he's also a reader in molecular neuroscience in Oxford at the Wellcome Trust Centre for Human Genetics. He's head there of the molecular neuroscience group. His laboratory carries out research into molecular mechanisms that underlie human speech and language. Simon is also the Isabel Lang Fellow at Oriel College in Oxford, where he teaches biochemistry and genetics to undergraduates studying medicine. So lucky undergraduates to be getting it straight from the horse's mouth. The Francis Crick Lecture was awarded to Dr. Fisher for groundbreaking research on genetics, evolution, molecular biology and neurobiology of human language. Ladies and gentlemen, I am delighted to have the great honour of presenting Dr. Simon Fisher to you.
Thank you. Thank you very much. I should start by saying that I feel deeply honoured to have been chosen to give this year's Crick Lecture. Francis Crick was an extraordinary scientist and a truly inspirational figure. In Matt Ridley's excellent biography, he describes how Crick made a mental list of the key scientific mysteries that he sought to solve during his career. And this list was surprisingly short. In fact, it included just 2 items. And the first of these items was life. And I think it's safe to say, actually, that Crick didn't do too badly in this regard. Not only did he, together with Jim Watson, uncover the double helical structure of DNA, but he also went on to decipher the universal genetic code that underpins all life on our planet, represented by this figure here, showing how this information encoded in DNA is transformed into sequences of proteins. So these 2 images here that you see represent the foundations of modern genetics and molecular biology. And they're still having an enormous impact on research today. The second item on Crick's list was no less ambitious, and it was consciousness. And Crick wanted to understand the very nature of the human mind. And so he proposed this hypothesis that your joys and your sorrows, your memories and your ambitions, your sense of personal identity and free will are in fact no more than the behaviour of a vast assembly of nerve cells and their associated molecules. And to restate this in a more explicit manner, he said, our minds, the behaviour of our brains, can be explained by the interactions of nerve cells and other cells and the molecules associated with them. So this was an unashamedly reductionist approach to life and to the brain. And he really felt that you could start to understand the behaviour of the mind, but you needed to look at the level of neural circuits and at the level of neurons themselves, and ultimately at genes and molecules. And I want to illustrate the power of that kind of approach today, but on my mental list I've got a different thing of interest, which is speech and language. And Darwin thought of this as an instinct to acquire an art. So the vast majority of children will develop complex speech and language skills effortlessly and without any need for formal instruction, so that by the time a child is 4 years of age, they already have access to a vast vocabulary, several thousand words, and they can assemble these words into complex, meaningful sentences, not only relating to the present, but also to the past and the future, and also to abstract concepts. And something else that we often take for granted is that then the child can communicate these ideas, convey these ideas, complex ideas, to others by this rapid, fine motor control of their articulators, of the muscles of the mouth and the tongue and the lips. And by doing this really remarkable feat, they pass on this information. And then this is matched by a feat at the level of perception, where we're able to decode this speech stream, and we're able to get an idea of what the person's actually trying to tell us. And another remarkable thing about language, speech and language, is the drive to acquire languages is extremely robust. So a deaf baby will babble with their hands, and groups of deaf children who have only limited access to sign language will go on to generate gesture-like systems of language that have many of the hallmarks of spoken language. So there's another reason why speech and language are of such interest to human beings, and that's illustrated by this slide. So you can strike up a conversation with either of these two organisms, but only one of them is going to give you an intelligible answer. And I'll leave that to you to figure that out. That's a very old slide, but it's one of my favorites. And it's probably the last time I'll use it. So it seems there could be something in our genes that helps build a language-ready brain. But before we can explore this possibility of a genetics of language, we need to think about some issues around the language of genetics. So what exactly is a gene? Well, you all know this because you read your newspapers and you watch BBC News, and you know that there's a gene for language, there's a gene for being gay, there's a gene for aggression, there's a gene for adultery even. Now this is all fine and dandy, but there's one big snag with this, and which is that there's no such thing as a gene for language. So geneticists like to talk about genes for this or a gene for that, and this originates from the early days of the field, in which a gene was described purely in terms of the dysfunction associated with its loss or mutation. So, for example, the gene for cystic fibrosis would be the gene that when it goes wrong, it gives you cystic fibrosis. Now, this kind of phraseology is still extensively used, particularly in the media, and that's because it's really effective at communicating the importance of genetic influences. So it's a useful thing, but it's not a real picture of what's going on at the genetic level. So what actually is a gene? Well, let's go back to basics for a bit. I'm sure that many of you are aware that DNA is a molecule that encodes genetic information, and it's in the form of a linear sequence of nucleotide letters, A, G, C, or T. And the genome that we so often hear about is the entire genetic makeup of an organism and the complete nucleotide sequence. And in the case of humans, this is more than 3 billion letters. And what I'm going to show to you in the next slide is this is not actually a blueprint. You hear a lot about genomes being a blueprint for life, but in fact, genomes are not blueprints. And chromosomes are the physical structures consisting of DNA and proteins which allow genetic information to be tightly packaged into the nucleus of every cell. Now, you've heard of mad geneticists. This is the reason why they go mad, because we spend all our time looking at these streams of sequence. Now, luckily for us, there are rules and people have figured out ways of actually decoding this, and actually one of those people was Francis Crick. So how to decode a gene? Well, what you can see here is this is DNA, the kind of double strand here, and it gets copied into something called messenger RNA, which is basically just a copy of the gene sequence. And it's identical, except that instead of T's, there are U's. But apart from that, it's just a linear copy. Now, this then gets read off by a special system of triplets of letters correspond to different amino acids, and there's about 20 different types of amino acids, illustrated here, methionine, serine, alanine, and they get strung together into a polypeptide chain. And basically, the sequence of amino acids here is just a copy of the information in here. It's a code that's been set up in the DNA. So the linear sequence of DNA gets converted into a linear sequence of messenger RNA, gets converted into a linear sequence of amino acids in a polypeptide. And then the polypeptide does this, which is to fold up. And the important point here is that when it folds up, it forms a 3D shape that determines what its function is. So the function of this thing is determined ultimately by the sequence of letters in the DNA. And that's really important to remember. So that's how linear sequence gets converted into 3- or 4-dimensional information. Now, as I said, genomes are not blueprints, and the reason for this is shown here. So genes, those kind of protein coding genes, have lots of spaces in between them. And for many of them, we don't know what all this other stuff does. But there are bits that are regulatory, and they control how these genes are switched on and off. So at the beginning of every gene, there's something called a promoter. And this promoter tells a gene whether it's going to be transcribed or not. And on top of this, when the gene gets transcribed, bits get cut out of the messenger RNA. And a lot of this, we still don't understand a lot of these kind of processes and why they happen. But these bits that get cut out are called introns, and these bits that get left in are called exons. And then not all of the mature RNA gets converted into a protein or gets used to direct the synthesis of a protein. There are bits at either end that don't. So much of the genome doesn't actually code for protein, and a lot of it we still don't actually know what it's doing. So these genes encode diverse proteins. They encode enzymes, channels, structural proteins, signaling factors, receptors. The shapes of the proteins can be so diverse that they can do all sorts of different things. And effectively, what we have is the instructions for building arrays of molecular machines. But on top of that, there's information that controls the way in which these genes are turned on and off in time and space. Genomes are very dynamic entities. And what this consists of are these kind of regulatory regions in the DNA and proteins that come along and bind to them and switch on and off this process of transcription. So if you're ever thinking about how a gene might map to a behavior, you've got to think it's got to go a long way to get to there. It's got to go through all these levels of complexity. So there's no straight line between this gene and this trait, such as language. So now I'm going to get back to the main topic of my talk, which is how does a geneticist get a handle on something like speech and language? So luckily for the geneticist, but not so lucky for the children, a subset of children grow up in language-rich environments but don't develop normal skills. So sometimes there's an overt cause, such as cerebral palsy or deafness or mental retardation, but often there's not, and about 7% of up to 7% of kids might be affected by unexplained developmental disorder of speech and language. These kind of disorders often cluster in families, and twin studies consistently indicate that there's a high heritability, and what this means is that there's a lot of role for genetic factors in predisposing to these kind of problems. But the problem is, how do we actually know which genes are causing the problems? So how do we get into the human genome and to see what the specific genes are that might have an impact on this? And here we have to just be lucky, really. And this is a family known as the KE family. And what you need to see here is that there are 3 generations affected. This is basically a family tree. And these squares are males, these circles are females. And if there's a line through it, it just means that the person had died before we made this figure, really. And what you can see here is that in these 3 generations, about half of the individuals in the family have this black shading, which indicates that they're affected with a severe developmental speech and language disorder. When a geneticist takes a look at this, he jumps up and down with great excitement, because this is just a classic example of a family that looks like it has damage to just a single copy of a single gene. So it's a dominant pattern of inheritance. If you inherit this dud copy from your parent, then you're going to have a problem. But there's no clue from just looking at this family what's actually wrong with them. Now, this family's been studied for about 20 years by Professor Farina Varga-Kadam and her team at the Institute of Child Health. And they've explored the phenotype, the profile of problems that the family have. And I want to just, if I can, play you. Hopefully this will work. These are children from the K family. So I hope you could hear there that these were kids from the K family, and you could hear that they have a severe problem with their coordinating their speech output. And I'm just going to play it again because with a transcript that Farina provided. And this is, I'll take you through it just so that you get a better understanding of actually what they were saying. Okay, so there's a kind of technical term for what the problem that they were having there, which is developmental verbal dyspraxia. So what they have is difficulties in the learning and production of these sequences of mouth movements. And remember, I said that this is one of the most demanding kind of rapid movement tasks that we do, is to coordinate speech output. And they don't seem to have similar problems at the level of limb movements, of limb praxis. So there seems to be something going on with the orofacial system and with controlling the muscles of them. And this shows you data from a comprehensive study by Kate Watkins and colleagues, where the kids are asked to repeat back pronounceable nonsense words. And here's unaffected kids from the family, and here's affected individuals. Here's affected individuals. And this is repeating back simple nonsense words with increasing numbers of syllables, and this is the number that they get correct. And this is complex nonsense words. And when I say complex, it means that there are these clusters of consonants which make them quite hard to say. So an example would be something like contrampanist or perplistronk, and I've been practicing those. So, and the point is that these kids will never have encountered these words before, and they have to listen to them and then repeat them back. And what you can see is that these are unaffected people from the family, and there's a big dip in the affected people. And these are adults who have had brain damage to the left side of their brain and have expressive aphasia, a kind of language disorder as a result of brain damage rather than during development. And you can see that actually the kids are doing very similar to these adults. And what you see is that the biggest hit comes from when you have these long syllabled words with complex articulation. So this is kind of characteristic of verbal dyspraxia. So it's safe to say that this is not their only problem. They have a whole host of other problems with language, and it doesn't express just their spoken language. You'd expect they have problems with speaking, so they will have problems with expressive language and with their spoken language, but it also affects their written language, and it affects their reception of language. So, and I just want to give you some examples of a couple of tests that are interesting, just to give you a flavor of some of the things. So here's a grammar test where somebody's shown this creature, and they say, this is a wug. This is a creature they've never encountered before. And now there's another one. There are two of them. There are two, anyone? Wugs, right, good. Just seeing that you're paying attention. So you can take the rule for plurals and add it on the end. and come up with the fact that there should be 2 WUGs. And there's all sorts of different rules that we use, and this just gets at one of them. And some linguists have claimed that the KE family affected members had a selective problem with this, and it seems that actually they do have problems with this kind of test, but they have problems across the board with other tests as well. Another kind of test that I'm just going to keep you on your toes. So this is a test that doesn't actually involve any expression of language or any reading or anything like that. So what you can see here is just 4 pictures, and then the child's given the sentence, which is the pencil on the shoe is grey, and they have to break that up and be able to figure out the relationship between these different things, and then figure out that this pencil on the shoe is grey, and of course you all know that it's this one, I hope. So, and if anyone's had any problems with either of these tests, then we'll be sampling some DNA from you at the end of the lecture. Okay, so there's been a lot of debate actually in the literature about how these different aspects of the disorder relate to each other. So some people think that having this kind of profound problem with learning how to speak can then impact on other aspects of your language development, and so you can have problems, these other grammatical problems as well. And other people think that maybe there's a core deficit that affects all these things. So this has been debated, and I don't want to spend too much time on it. But what is clear is that there's some anomalies going on at the level of the brain in these kids. So I want to very, very briefly, this is kind of a 1-minute guide to the neural basis of language. So bear with me a bit. So a kind of classical, this is your brain, and it's the outer part of your brain, it's the cortex. And what you can see here is that classically there are 2 regions of the cortex on the left side in right-handed people called Broca's area and Wernicke's area, and they form a circuit with a white matter tract between it called the arcuate fasciculus, which sounds like a difficult nonsense word, but and these regions have been linked to different aspects of language function. So Broca's area, for instance, has been linked to speech production and grammar, while Wernicke's area has been linked to comprehension and other aspects such as that. But actually what's emerged, and a lot of this was based initially on studying people with lesions in the brain and studying which bits of their brain were damaged. So there's a form of aphasia called Broca's aphasia and a form of aphasia called Wernicke's aphasia. But what's become clear over the years with more and more study is that actually many different parts of the cortex are involved in when you're speaking and when you're reading and using written language. And this is just to show you that you're using a lot of the circuitry. And also it's become clear that these kind of different areas all do a bunch of different things. They're not exclusive for one particular part of language function. But it still holds that these are still very important areas. So we've got to move away from understanding just these are kind of little blobs in the brain to understanding neural circuits. And it gets even more complex than this, because this is, if I took your brain and kind of sliced it in 2, this is just a kind of cross-section. And this shows you this is the cortex on the outside, which is all folded and does a lot of the big things that make us human. But underneath this are other structures. So here is something called the basal ganglia, where in there you've got the striatum, the caudate, and the putamen. And it seems that damage to these areas also will give you kind of language problems. So there are circuits that involve the cortex and these kind of basal ganglia structures that affect your ability to use language. And also the cerebellum, which is actually a very old, evolutionarily old kind of structure, has been implicated in language processing as well. So we need to move away from these kind of focal kind of areas in the brain to consideration of circuitry. And what this shows you is again, data from Farriner's elegant studies of the family over the years. And what you can see here is some data from Kate Watkins' paper in Brain, where the family were looked at the level of neuroimaging, where they can look at the structural features of them, and the affected individuals were compared to the unaffected individuals in the family. And what these are showing you is if you take small units, divide the brain up into not pixels but voxels, kind of 3-dimensional pixels, and compare in the affecteds versus the unaffecteds how much grey matter density there is, how much cell density there is. You can see there are differences, and this just shows you some of the differences that are seen. So in the affected members of the KE family, they have a reduced grey matter density in this caudate nucleus, which is part of the striatum that I just showed you. And they also have reduced grey matter density in the left inferior frontal cortex. Again, this is near to the Broca's area. And also remember, this is a motor disorder, so there's also some involvement of the supplementary motor area. So what I want you really to take home from this is that there are subtle abnormalities in the structures of their brains. They're not really dramatic, but they're significantly different. So what does this mean at the level of function? So this is from a paper by Frédéric Ligeois and colleagues from a few years back. And what they did was to give the kids in the family a task where they're given a noun and they have to generate a verb. And what you can see here is that if you do this, and what I should say is that they're just thinking their answers in this. They're stuck in an MRI scanner, and they don't actually have to produce a vocal output. So they're just thinking about a verb that's associated with a noun that they're provided with. And what typically this activates in unaffected people, and in these unaffected members of the KE family, is you get activation on the left side in this region that sort of overlaps with Broca's area. Now, if you put the affected kids from the family in the scanner and you do this same kind of task, what I'm going to show you now is a kind of averaging from a number of different individuals. So it's not like they all activate these regions, but you see some striking differences. So what you can see here is actually that there's underactivation in this region that around Broca's area that was originally activated. And in fact, now you're seeing bilateral activation on both sides of the brain in all sorts of different places. And again, the take-home message from this, perhaps, is that their brains are doing something different. And it's not necessarily about the way that their muscles are working. There's something going on at the level of the brain that's influencing how they're able to process language. And also, I should say that when they're given this in a kind of overt thing where they are actually they are actually doing the task and giving speech output, then you see similar results. But the point is they are able to do this task. It's not that they can't do it. OK, so now I want to sort of move on to really where I come in, which is to look and see where the gene is that's damaged in this family. So about 12 years ago, when I was working in Tony Monaco's lab as a postdoc, we started working on the KE family, and we were able to map the gene in the family to an area on this is chromosome 7. So we mapped it to a region here, and within this region, what we knew was that there was some kind of damage to the DNA. We didn't know what it was or exactly where it was. And there were about 70 or so genes in there. And working with Cecilia Lai in Tony's group, we started sifting through these genes and trying to find which was the damaged one. And then I got a phone call from Jane Hurst, who was a clinician who'd originally actually seen the KE family and wrote one of the first reports of the KE family's problems. And she pointed out a new case that she'd come across, which was a kid called CS, and he had a very different type of genetic change. He had something called a translocation, and what you can see here is kind of a spread of his chromosomes made by Cecilia. And here you can see this is his normal chromosome 7, and there's a breakage here, and it's got stuck onto chromosome 5 here. And over here is chromosome 5, and with a breakage here where it's attached to chromosome 7. So he's actually had a big breakage, but all the genetic material is there and it's intact. And over here is his normal chromosome 7. And at this point here, there's a breakage splitting a gene. And the point is that although he's not related to the KE family, he has a very similar type of disorder, a kind of speech and language disorder that's made Jane Hurst think, well, this is actually very similar. And the interesting thing was that when we looked to see where this breakage was, we actually found that it was mapped in this region here. And in fact, when we zoomed in, we could see that actually it broke a gene, slap bang here. So then we went and looked at that gene in the KE family and the affected members, and we found that there was a single letter change to the DNA. just at this point here. So the affected individuals, instead of carrying, normally you should carry a G here, instead of carrying a G, they carried an A. And the reason that you see two letters here is because they have two copies of the chromosome, one from one parent and one from the other. And there's a G on one copy and an A on the other. So this is, and then I should say that all the affected members in this family carried this mutation. None of the unaffected members carried it, and nobody that we then looked at a large number of controls in the general population, and this mutation has never been seen since. So it seems to be something unique to the KE family. So what we can see here is that we have this mutation here in the DNA, and we know that there's this behavioral deficit over here. And maybe this can tell us something about how these genes map to behavior and in this disorder. And maybe that will actually tell us something fundamental about how DNA can influence something as complex as speech and language. But if you were paying attention at the beginning of the talk, you realize that I said there's no such thing as these kind of direct arrows. Okay, so we have to forget about these arrows. But what I'm going to try and do is fill in the gaps in a different way. Okay, so one thing we can do is push back from the behavior level and look at the brain. And I already talked about that, and I showed you these beautiful results from Farriner's work. And so that makes life a bit easier for us because we only need to get to here, effectively. But we still need to go in this direction. So can we do this? Okay, so now we kind of are lucky because this mutation that we see in the KE family occurs in one of these protein coding genes, and it occurs slap bang in the middle of the protein coding region. And we can, with the help from our friend Crick, we can translate the sequence of this gene into the protein sequence. And what you can see here is this is a stretch of sequence, and this is where the mutation is. Okay, so it changes a particular amino acid in the protein from an arginine to a histidine. This is this R to this H. And in fact, we can even make some predictions based on previous studies of proteins that look similar to this. And we can show that actually this part of the protein that we're looking at would normally form this kind of structure, which is called a winged helix. And what you see here is two helices and then a third helix here. And this is where you have this mutation. So how does that get us any closer to understanding what's going on in the KE family? Well, this stretch of sequence that I've just shown you that forms this interesting structure is called a fork-head box. And there are a whole series of different genes in the human genome that make these kind of fork-head box proteins. In fact, there's 43, and they're divided into different subgroups for fairly arbitrary reasons, and they're given letters from A to R. So this gene is FOXP2, because it happens to be the second member. When we found it, it was the second member of the P subgroup. So it's FOXP2. The fork-head box is a part of the protein with this 80 to 100 amino acid stretch, and it allows it to stick to DNA. So here comes Crick again. Here is DNA double helix in purple, and here is that part that I just showed you before in a different schematic. And what happens is these two helices position this third helix so it gets stuck into the major groove, into this big space in the DNA. It sticks to DNA. And what's interesting is that this particular mutation, the KE family, what we've shown is that when this gets changed to a histidine, it can't stick to this DNA. Why is this important? Well, it actually gives us a big clue to the function of the gene. So what this gene does is it makes a transcription factor. So FOXP2 gene itself gets transcribed into messenger RNA, and you get a protein called FOXP2 protein. And what the FOXP2 protein does is it goes to target gene X or target gene Y, I'll show you in a second, but it can stick to a promoter and activate transcription, which means that when transcription gets switched on, the protein gets made. So when FOXP2 is on, you get activation of this downstream target. So it can regulate the expression of other genes. Alternatively, a gene that's normally switched on can be switched off, repression, by FOXP2 binding to it. This is a kind of master regulator gene. And what you should remember is that in the KE family, they have one normal copy of FOXP2, but they also have one damaged copy. So here's chromosome 7. Again, remember, you have two copies. And one of their copies is making normal FOXP2, and one of their copies is making dud FOXP2. It's making damaged FOXP2. So we think that the reason that they have a problem is because they don't make enough functioning FOXP2. So what I'm showing here is if we took the whole of the FOXP2 protein, which includes not just this fork head box, but a whole bunch of other regions, we took the folded protein, and we kind of stretch it out into a line. And it has a kind of start and an end here. And what you can see is just a bunch of different functional regions doing different things. There's the fork head box domain. There's where the missense mutation is in the KE family, which is changing this arginine to a histidine in the amino acid sequence. Remember, I told you there was this other kid who had a translocation breakpoint, which was just inactivating the gene in a different way. And the point here is that if we want to find other mutations in other children, we have to look at the whole gene. We have to screen the whole gene because the mutations could be anywhere in this protein. So we did this for about 50 kids who had similar speech and language problems to the KE family, and we only found one mutation out of 50 kids. So it's a very rare thing to find a mutation in this gene. But actually, it was a great mutation to find. It's one of the geneticist's favorite mutations because it's called a nonsense mutation. And what it does is instead of changing an amino acid at the position in the sequence, it inserts a kind of stop sequence. It says, stop making that protein. So what that does is it gets rid, it makes a shorter protein that lacks all these important functional domains. And we were just looking at individual kids, but it turned out that this kid had a sister who had similar speech and language problems and a mum with a history of problems as well. And they all had this mutation. And what's worth remembering, or what's worth emphasizing here is that, again, they have one normal copy and they have one damaged copy of FOXP2. So it seems that reduced functional dosage would apply here as well. OK, so we've been able to go from the DNA a bit to the protein. So we know that if, with this mutation in the DNA, it changes the way that it actually inactivates the way that a protein works, and that protein is FOXP2, and that protein is a transcription factor, which switches on and off other genes. But the problem here now is that proteins and genes don't act in isolation. They don't act in a vacuum. They act in cells. So what can we say about looking at FOXP2 in cells? Well, we can take these are human neuron-like cells that we grow in the lab. And what you can see here is that we can label the nuclei, the middles of these cells where all the genetic material is, in blue. And then we can make the FOXP2 glow in green. And if you look here, normally FOXP2 likes to be in the nucleus, the protein. The protein sits in the nucleus because that's where it's sticking to the DNA of other genes, switching them on and off. Now, if we put this is the version that's found in the KE family, and this is the version that's found in that other family that I showed you. And if we put those into these cells, what you find is, say, sometimes, not always, but sometimes, in the case of the KE protein, it doesn't get into the nucleus. And this version, that's a very shortened version that you find in this other family, never gets into the nucleus. It's always sitting around in there. So again, this is interesting, but it doesn't really get us any closer to understanding the function of the gene and of the protein. So now I want to go on to something else we can do with these cells, which is a bit more interesting. And what we can do here is to take the cells, and we want to know what are the downstream targets that FOXP2 is regulating. And the way we can figure that out is by finding where it's binding in the genome. What DNA is it sticking to? And what you can see here is this is just a little stretch, not to scale, of DNA from some cells, and with FOXP2 and some other transcription factors, other proteins bound here. And what we can do is we can cross-link these cells so that basically we're just sticking the proteins to whatever DNA is around. So they're naturally stuck now to where they would normally be binding. Then we can we can purify the DNA and we can cut it up. And then we take an antibody that likes to stick to FOXP2, and we throw it in, and we pull down FOXP2, but it's still stuck to the DNA that it normally likes to stick to. Then we reverse the crosslinks, we purify the DNA, and what have we got? We want to see what we've got. Now, what I should say here is that we get a big mixture of lots of things, some of which can be false positive results, but the great part is we end up with a whole series of pieces of DNA that are likely to represent downstream targets of FOXP2. And this is work that was done by Sonia Vaness in my lab, and one of the first genes that she came across, one of the first regions of DNA that was found happened to also be on chromosome 7. This is just a coincidence that FOXP2 is on chromosome 7, and so is this other gene. And it has this rather annoying name of contactin-associated protein-like 2, which is a difficult nonsense word for kids to say. But, and so I'm going to call it catnap2 just for a shorthand. And this gene is bound to by FOXP2. What does this gene do? Well, this makes a protein that likes to sit in neurons. Here's a picture of a schematic of a neuron. The neuron has a big cell body here, and then it has an axon that sends down these signals to the ends here. And what this protein that this gene makes does is to sit around these regions of the axon, where these are surrounded by these myelin sheaths, which make communication much faster. And what this protein does is it likes to sit around here and bring special potassium channels to these regions. And it seems that it might help the conductance of nerves in the central nervous system. But what's also interesting is that it likes to sit in the membranes. It's also got the name Caspar-2 just to confuse things further. But here it is. This is the protein of this target. And it mediates, it kind of communicates from one neuron to other cells during development in the developing brain. So it helps to set up the brain, if you like. So it does a lot of different things, but it's a very interesting gene. And what we found was that, in fact, not only does FOXP2 bind to it, but you can show if you take cells and you upregulate FOXP2, then you get downregulation of this target. We could also show this. This is work that was done by Brett Abrahams and Dan Geschwind in Dan Geschwind's lab at UCLA. And what you can see here is where is this gene switched on and where is this gene switched on in the developing brain of a human fetus. And actually, all you want to get from this rather complicated slide is that there's a strip here of where this catnap2 gene is switched on. And here's the strips where FOXP2 is switched on. So it seems where FOXP2 is particularly high, you have low levels of contactin-associated gene, and vice versa. So again, it looks like it's switching down the gene. So why is it interesting to study these kind of targets? So I've already shown you that FOXP2 disruption causes an unusual, severe speech and language disorder, and that mutations are pretty rare. And in fact, if we look at typical forms of language impairment, which affect up to, as I said, 7% of kids, we don't find any FOXP2 mutations. And these kids don't normally have the kind of severe speech problems that the KE family have that you've heard. So Diane Newbury in Tony Monaco's lab investigated this contactin-associated gene, which is this downstream target of FOXP2, in children with common forms of language impairments. And what she found was that these variants, certain types of variants in this gene, were associated with poor language performance. So kids that carried these so-called risk alleles did more poorly on tests of, for example, nonsense word repetition here. And remember, this is the kind of test that I was talking about earlier with the KE family, where you have to repeat things like contrampanist and perplistronk. But these kids don't have this kind of overt speech problems, but they still have more subtle problems with nonsense word repetition. And what you can see is you can carry, again, you have 2 copies of every gene. People who carry naught do kind of OK on this non-word repetition. And as you have increased numbers of risk alleles, it's bad news for your nonsense word repetition. It reduces it. Now, this is quite different from what we saw for FOXP2, because this is certainly not the only gene that's important in these kids. So in this case, it's like they have to have damage to several different genes, multiple different genes, to be able to get language impairment. I should say that these families were 200 families collected by the Specific Language Impairment Consortium, a UK-based consortium that looks at these kind of disorders in kids. And so this is very different from FOXP2, but it gives us a new insight into what might be causing problems in language impairments. And the other thing that was interesting was that previous studies had shown that if people carried these similar kind of variants in this gene, they were also at risk of getting autism. And in fact, it was correlated with delayed language in autism, in the onset of autism. So this is a very interesting finding. And what I think it shows you is that actually, just by studying at this level of the cell and at the neuron, just culturing these cells in the lab and looking for downstream targets, we were able to get into a new piece of biology and a new interesting piece of information that was relevant to the medical situation, because we were able to show that it was important for kids with SLI. Okay, but there's still a problem here, which is that cells and neurons don't act by themselves. They act in the context of neural circuits. Neurons wire up to each other. I mean, so what can we say at this next level? Now, here we run into a problem, because ideally what we'd like to do is be able to study in the humans the neural circuitry. But unfortunately, parents get very, very upset if you try to take out their kids' brains when they're too young to study them. So we have to get around this by jumping to a different species. And the species, the favorite species of the geneticist is this one. Now, by now, some of the sharper members of the audience will have recognized there's a problem here. So does this mouse have a speech and language disorder? And unfortunately, outside of the research labs at Disney and Pixar, rodents don't tend to talk. So there is a serious point to be made here. Why should we jump to the mouse, and is this going to tell us anything of any use? Well, actually, it will do, we hope, or we think. And the reason is, remember I took the protein and I stretched it out, and it's 715 amino acids long. And here is the protein in humans. And in mouse, it's 714 amino acids long. And what you can see here is that actually, I'm showing you here the differences between a human FOXP2 protein and a mouse FOXP2 protein. And basically, there's not many, right? There's three differences where there's one amino acid instead of another. And here you have something called a glutamine tract, a tract of cues all repeated after each other. And there's a slightly shorter stretch in mouse, but nothing particularly dramatic. So effectively, it's a very similar version in mouse. And in fact, all vertebrates have a version of FOXP2 that looks very similar. And not only that, but if you look in the brains of a human and in the brains of a rodent, and you can ask, where is FOXP2 switched on in the brain? Remember, I talked about the cortex and the striatum and all these different regions. Well, actually, the genes switched on in similar bits of brain in the human and in the mouse, and in other species again. So this makes sense for us to be able to look and try and understand. Oh, and I should say also that these regions that we've talked about overlap particularly the striatum, where you saw that they had a reduced grey matter density in the KE family, and also areas of the cortex. These regions and structures overlap. So the places where FOXP2 is switched on are the places where we're seeing things going wrong, or Farron is seeing things going wrong in the KE family. So this is very encouraging. And some things that we can look at in the mice that we can't... sorry, before I go on, I should say that here's the mutation in the KE family, the R553H missense change. And here you can see the equivalent mutation in the mouse, and it's called R552H, and that's just because it's slightly different length of protein. But other than that, it's exactly the same change. So we've made a mouse that carries exactly the same changes in the KE family. And what we can look at is the mice that carry one normal copy and one damaged copy, just like in the KE family. And what we're interested in looking at in these mice is really the circuitry. And this just shows you a schematic of neural circuits. So there's neurons, and they put out these axons, and axons stick to other neurons and send signals to these other neurons. And what you can see here is that the other thing that we can be interested in is when electrical signal gets sent down an axon, it eventually gets to this thing called the synapse, which is a kind of chemical region where the electrical signal stops, and it has to jump across the synapse. And it does this by sending out some molecules over here. And the key thing here is that the way that our brains learn, much of the way that our brains learn is based on changing the strength of these synapses. It's based on whether a signal, when it gets to here, jumps across to the next neuron. And these signals get changed in response to learning. So for instance, if a neuron receives a lot of stimulation, it might remember that and then be ready to
get more stimulation and be kind of activated, and that's called long-term potentiation. And another thing that might happen is that the neuron might be depressed by lots and lots of stimulation. So the question is there anything going on at these kind of levels in these mice that we've looked at? And we haven't been able to really see much in the way of the kind of wiring, but when we've looked at this kind of process here, which is sometimes termed synaptic plasticity, we see something quite interesting. And this is work that was done in collaboration with Rui Costa at the National Institute of Health in Washington. And what you're going to see here is if we take a part of the brain where we know FOXP2 is expressed and we know that it goes wrong, or we know that we see functional abnormalities in the KE family, so this part of the striatum, and we can take cells and we can, or circuitry, and we can stimulate the circuits. And what you can see here is this is the normal responsiveness of the circuit, and then you give a high-frequency stimulation. This is in normal mice. And the neurons kind of remember that they've been stimulated. So this is called long-term depression. So they stay, if they receive any more signals, they just kind of stay quieter than they would be. And this is what happens if you take the striatal cells from a mouse that has this KE mutation, one damaged copy, one normal copy. And what you see here is that initially it looks like it's going to remember, and then it goes straight back to baseline. It's like these circuits just don't know what they're doing. They can't learn in the same way. This is a very striking phenomenon. So there we've got something at the neural circuitry level in the mouse. But of course, we've still got a problem because we're studying a mouse, and mice don't speak, as we've kind of dealt with. So do we know that this really has anything to do with anything? Is it actually doing anything at this level? And this is where we have a kind of a nice gym for mice, which was developed by some colleagues, Joe DeBono and Keith Channon, and actually at our institute. And this was actually developed to look at cardiovascular training, but it can be used for other things as well. So what you see here is a kind of tilted running wheel, and you can put this into a mouse cage, into a home cage environment with a mouse. And mice are kind of fitness freaks. So if you shove one of these things into their cage, when the lights go out in the mouse house, they all leap on top and they run around much of the night. So they love to run on these things, and this is linked to a computer and it records all the activities. So this is voluntary. The mice do absolutely as much running as they like. But what's curious is because this is a tilted wheel, it's a bit tricky for the mouse to learn to run on it. And it takes it a few days after you've stuck this in the cage for the first time. It takes it a few days to figure out how to use it and how to run fast on it. And we can look at this kind of motor pattern learning by looking at what we get from the computer output from, and we're not going anywhere, we're not actually picking up the mice at any point here. So it's a really nice, non-invasive kind of assessment of what's going on. And what we found was that mice that carry one damaged copy of FOXP2, they have normal baseline motor behavior. They can run okay and do all the normal functions, but they show deficits in learning on these kind of wheels and on other ones. And what's interesting here is this has nothing to do with our facial system, right? This is just a kind of limb. So how this relates to what's going on in speech and language remains to be shown. And some of you might be asking, okay, well, mice do squeak, so perhaps there's a squeaking disorder in these. And in fact, some people who've studied mice that have damaged FOXP2 do claim that all that they've looked at so far are pop vocalisations, which I'm showing here. But there are some claims that FOXP2 might be important for vocalisation in mice. I should say that we haven't really seen much evidence of that. But what I want to show you here is that mouse pups produce these three types of calls. And one type of call is just the kind of call that you normally would hear, like this audible squeak. And another type is this ultrasonic vocalisation here, which we can't normally hear. So it's outside our range. And they also make these little click sounds like. So what you can see here is mice that have no FOXP2 at all, and they're still making these sounds. But these kind of innate pup vocalizations are not maybe hugely interesting, but what could be more interesting is that young adolescent male mice, when they're exposed to female urine or to females themselves, they get kind of excited and they start to sing. And we can't normally hear this because it's in a very high range. But Tim Holy did this clever trick where he shifted the pitch down so that it was in the audible range. And here it is. Here is just a segment. I'm going to play it to you. So that sounds a little bit like birdsong. And this is a very interesting thing to try and to unpack and to study, because we can look at this level and see whether, as you can see, there's different types of syllables and their sequence. So we could maybe look in the mice that have these KE mutations. And here you can see this was done in collaboration with Günter Ehret's group and Ulm. And you can see that here there are these complex vocalizations that these mice are making. And this is the male mouse, and this is actually during mating. This is the female mouse, and these are called defense calls. I'm not quite sure why that is. But anyway, so that's mouse song, but it's a very, very unexplored kind of area. And what's much better studied is songbirds. So a lot is known about how birds learn songs. And now I'm going to show you work that's not from my lab at all. I had no involvement with this at all, but this is such a beautiful story that I really had to show it. And this is from work by Constance Schaaf's team in Berlin. What you can see here is zebra finches, they learn their songs. So after they're hatched here, there's an initial phase of sensory learning and then sensory motor learning where they're kind of producing this. Initially, they produce something called subsong, which is very quiet little murmuring. And then, in response to an adult tutor who they're hearing, they start to try and mimic this song. And they gradually get better and better at it. So they go through this plastic song stage. And eventually, they get crystallized song at day 90 after hatching. And then the question is, are they correctly imitating this song that they initially were given? And interestingly, they can get to about 100% copy success here. So they can really, you can see here is a sonogram of a zebrafinch song, and here is the initial one. And what you can see is that this is a great example of vocal learning, and it's a bit like what we do. I mean, it's not exactly the same, obviously, but it's a nice model to be able to study at least some elements of what might be important for the processes that we use. So they listen to something, and they practice, and they learn to kind of say it just through this kind of learning. And there's one area in the brain, and it's actually in the striatum again. So again, the striatum pops up, which is called area X, that's already known to be really important for this kind of learning. Again, it's part of a circuit, but it's known to play a really important role. And that's an area that in songbirds shows quite strong expression of FOXP2. So what Constance's group did was they injected a lentivirus into this one little part of the brain and used it to knock out FOXP2 expression or knock down FOXP2 expression. So this part of the brain now didn't make as much FOXP2 protein. And remember, FOXP2 is very similar in these other species, so we're talking about a similar version of FOXP2. Excuse me. So I'll play you a few little snippets here. And this is if you put a virus, but it doesn't go into this area X. So this is kind of what a normal tutor song sounds like. Okay? And this is the pupil who's copying it pretty well. This is if you've knocked down... Sorry, this is the non-targeting, a virus that's not targeting FOXP2, but it's going into area X, so it's a kind of control. This is now the one where where we're going into an area that's not area X. So again, quite accurate copying. So now, these are two quite nice examples of what happens when they have FOXP2 knocked down. That's the tutor. And listen to what the pupil does. It's a little bit different. And what's happening is that it's missing certain types of syllables. And in addition, it's not copying the syllables as accurately. And this is one of the most striking examples that Constance has. That's the tutor. That's the pupil. So they're missing syllables, and they're not, and they did a complex analysis to really show statistically that this is the case. So that's a very interesting way to go. So basically, we've still got lots and lots of gaps in here, but we've got a kind of framework of different levels, and there are tools that we can use to find out more. So, for instance. Catherine French in my group is knocking down FOXP2 in the mouse brain, knocking out FOXP2 in the mouse brain in individual circuits to try and figure out which circuits are most important for these kind of motor learning problems. But it's not the gene for speech, FOXP2. I need to emphasize this. So Versions of FOXP2 are found in highly similar form in vertebrate species, a whole range of different vertebrate species that don't have speech and language. I don't quite know what that species is, actually. So we think maybe it had an ancient role in the brain of a common ancestor. And FOXP2 is not just working in the brain, it's also switched on in parts of the lung, intestines, and heart. It's doing different things in different places, and this is what regulatory genes do. They get reused, they get recycled. You don't want to evolve a new protein, a new gene to do a new thing. So it's not the speech gene, but I want to finish off by placing this in the context of human evolution. So this, I've called it a revenge of the speech gene. Remember I said that a human and a mouse have very, very similar versions of FOXP2. Well, if you take a chimp and you compare a chimp, the chimp actually looks a bit more like the mouse. And the human version of FOXP2 has these 2 amino acid changes that have popped up in recent human evolution. So when it comes to FOXP2 protein, a chimpanzee is more similar to a mouse than it is to a human, which is a curious finding. And this was work that was done in collaboration with Svante Pääbo's group in Leipzig. And what they showed was that if you looked at, this is the mouse, and then these are different primates, and this is about 70 million years ago when there was a common ancestor to the mouse. And what you can see is that the first number shows the number of changes to FOXP2 that has an impact on the protein sequence. And the second number is the number of silent changes that don't do anything. So some changes won't actually affect the protein. So in these 70 million years, not a lot's happened on the mouse lineage, just one significant change on a very long period of time. And then here's the primates. And again, not much happened. There's one change on the orangutan. And this was about 5 or 6 million years ago that we split from the chimpanzees. And there's been these 2 changes that I showed you before. And Svante's group went on to study this in more detail, and they could show that actually there was evidence from looking at other bits of the genome around these changes of Darwinian selection, so of the gene, of this new variant spreading. So an accelerated change of FOXP2 on the human lineage after splitting from the chimpanzee and evidence of Darwinian selection. What does this mean? This does not mean that FOXP2 was the driving force behind the evolution of spoken language. For starters, who would the person who first had the new variant, who would they talk to? So remember I said that this gene probably has an ancient role in the vertebrate brain, and maybe it's involved in supporting how we make sequences of movements. So when spoken language was emerging, perhaps these modifications or some other modifications to FOXP2 would have given people advantages in vocal communication. So if spoken language was already emerging for whatever reason, and this version would have spread. So it's just one piece of a complex jigsaw, but it's an interesting piece nonetheless. And what's been difficult is to figure out when these changes actually occurred. What Svante's group initially did, and another group did this as well, was to take modern humans, look at FOXP2 in modern humans, and try and figure out when in history these new variants had arisen. And they, using this, they fairly robustly found that it was probably within the past 200,000 years, which is really recent and coincident with the emergence of modern humans and modern language. But then they went ahead and they looked at Neanderthal DNA. It's very hard to sequence Neanderthal DNA. It's all broken up, but Svante's doing it. And when they looked at this, the Neanderthal DNA should have had the same version as the chimp, because Neanderthals split off from us maybe around 300, 400,000 years ago. Certainly not as recently as 200,000 years ago, maybe a bit further back. But what the Neanderthals showed was that Neanderthals carried the same version as modern humans. So the question is, did these happen recently, or did they happen some way further in the past? And I have to say, we don't know the answer to this question at this stage, and people need to look into it in more detail. So, to bring you back to reality, I've shown you that missense and nonsense mutations in FOXP2 cause this severe speech and language disorder in humans. And FOXP2 is a regulatory gene. It switches on and off other genes, and its downstream targets offer entry points into neural pathways that are involved in speech and language, such as this CatNap2 gene. And I've shown you that versions of FOXP2 are found in many species in very similar form. And in mice, it seems to be important for motor skills, and in songbirds, it seems to be important for vocal learning. So our unique capacity for complex spoken language hasn't just popped up out of nowhere. It's built on these evolutionarily ancient systems And this is, FOXP2 seems to fit into this kind of framework. And what I hope the main point I've shown you is that this, by studying FOXP2, this is kind of paradigm for how we can try to bridge gaps between genes, brains, and behavior. And I want to highlight, out of the people who I've collaborated with on FOXP2, in particular, Tony Monaco, who was my mentor for many years, and Cecilia, who worked with me to find the gene, who I don't have a photograph of, but hopefully she's here somewhere, and Farina, who has done so much work on the KE family over the years. And then all these other people, many of whom I've mentioned as I've gone on. And I have this Royal Society, URF, and some funding from the Wellcome Trust. These are these other sources. And I'm currently looking for funding if anyone wants to give me some. And these are members of my group, ex-members and current members. And thanks to Professor Schwartzman as well from the Gary Larson Institute. And just for the last word, because I know I've kind of run over slightly, but the last word from Francis Crick, which is that he acknowledged that the single most characteristic human ability is that we can handle a complex language fluently. But he also noted that this ability leads to another strikingly human characteristic, one that is seldom mentioned, our almost limitless capacity for self-deception. So, and he went on to say, man is endowed with a relentless curiosity about the world. We must hammer away until we have forged a clear and valid picture not only of this vast universe in which we live, but also of our very selves. Thank you.
Thank you very much for that. We have time for one or two questions before I say a few words. There are two microphones circulating, so please wait until a mic reaches you. Okay, one in the middle here.
Thank you very much. Delighted. My name's George Crowther. I'm a psychologist. I'm not a speech and language therapist, but I guess there are probably speech and language therapists in the hall. And I suspect that speech and language therapists, as indeed many psychologists, will not have the kind of privileged information available to them that you've presented us with this evening. And what intrigues me about speech and language therapy is they are not in possession of this information. Even if they were, what difference would it make? And they're still going to encounter these children day in and day out with significant speech and language difficulties, and presumably they go forward the best they can according to their knowledge, skill and experience.
Well, I think we would hope that they could be supplied with some of this information. I think this is something we're trying to raise, firstly, awareness that things like specific language impairment are actually that genes contribute significantly. Now, this doesn't mean that you can't do anything about it. There's a kind of myth that genetics is deterministic and it means you can't actually make changes. And in the case of these kind of things, we would say that the earliest that one can identify a child as being at risk of having a problem, the earlier one can intervene with standard therapies. And that this is a great way of, hopefully, I mean, in the case of the FOXP2 mutations, we can diagnose that the children will have problems very, very early on. In the case of these other genes, where the links are a bit more complex, it's harder to do that, but I think we will be able to do that. The other thing we can do is maybe to help understand what different subtypes there might be of the disorder. People have tried to do that over the years by looking at the profile of problems that they have, but as yet they haven't been able to successfully divide them into groups that would have maybe more different types of therapies might be more effective. So I think this kind of information is important. I think we're trying to get it across to the speech and language therapists in an accessible way, and I think it will lead to changes, but it may be a while to come into practice.
Hi. You said that FOXP2 may have different regulatory processes in our body.
Yes.
I was wondering whether you found any sort of disorders in the K family as a whole, maybe to do with the other regulatory processes.
That's a good question.
And another question is, do you think that upregulating FOXP2 would solve their problem?
So the first point is that, as I understand it, they don't There are no detected problems elsewhere outside of the brain in the KE family. But the point there is that they still have a functioning copy of FOXP2, and it seems that knocking out genes has different effects depending on whether you completely remove it or whether you've just got some there. So it seems like part of the brain is particularly sensitive to the levels of FOXP2. With regard to your question, if you could upregulate it, would that remove the problems? I think the difficulty there is we're not sure whether this is actually a disorder of really early development or a problem in the functioning brain. So we're not sure if it could be fixed by changing FOXP2 levels, because it might be that the damage has already been done, if you like. But it's a very interesting question. Thank you.
Yes. Thank you.
My name's Kathleen Mains. I just wondered whether the family has been studied for their musical ability and their ability to construct and decode music, which is so closely related to language.
So this hasn't been done yet, but I gather Fariner is interested in doing it, so it's not known yet.
Any more? Okay, last two more quick questions. One there and then one here. Yeah.
Thank you. My name's Alan Sturt. What's the difference between neurons that interact with each other within the areas of the brain that you've shown on the screen, and the neurons which interact between those areas and other areas in the brain, because more than one is involved in any activity? I mean, as I understand it, they're all the same. So how do they know they are doing these different functions?
They're not all the same. I mean, the first point is that the brain is incredibly heterogeneous in its structure in terms of the neurons, and there's vast diversity in both in the shapes of the neurons, in the kind of genes that they're expressing, in the ways that they're wired up. Some of them express one transmit. You remember when I showed you the synapse, where the link between the end of one neuron and the input of another one, if you like. The kind of transmitters that go across these vary from different neurons to different neurons. So there is an enormous diversity in the neuronal makeup of the brain. And that's one of the challenges, actually. You can't just say, oh, well, I've studied this gene in a neuron in the lab, because actually the neuron that you would study grown in cell culture is nothing, it's not that similar to what you might see actually in vivo. But it's a good starting point. There's a lot of there's a lot of chemical variability in terms of that they express different profiles of genes within them. So there's diversity within that. And also what leads to the functional diversity on top of that is the way that they're wired up from one region to another. So it's like a complex circuit diagram. And so the impact that a neuron would have on a particular behavior is not just about what that neuron looks like or what genes it's expressing, but also about what other neurons it's connected to in different parts of the brain.
There was one more. Yes, there you go.
Hi. First of all, thanks for a very interesting talk. Nigel Wood. The question is whether there have been noticed patterns in the syllables that children can recognize or pronounce.
Sorry, I didn't understand the question.
Are
there particular patterns
of specific syllables or sounds
that
the children recognise?
So typically in verbal dyspraxia, there's actually a lot of variability. There's inconsistency, if you like. And this differs, say, from something like a dysarthria that would affect the musculature, in which there would be certain types of sounds would be made incorrectly consistently. So one of the characteristics of a kind of dyspraxia, of this kind of articulatory deficit that is the main thing that you see in the KE family, is this variability.
Okay, I think we have to stop the questions there. There's obviously a tremendous amount of interest in this topic. I want to thank Simon Fisher for an absolutely wonderful lecture and a fascinating story. I should have said earlier that the Francis Crick Award is for younger scientists, But I think the lecture we've heard would do anyone of any age a great deal of credit. We've heard a beautiful story going right from gene discovery to gene expression to association with a disorder. And it's, I think this is going to become a classic. It probably is already on its way to becoming a classic as an example of what can do with population, or with family studies tied into the modern techniques of molecular biology and so on. I think he carried the audience with him. I felt that we were with him all the way. It was delivered with style and with humour, and for that we are very grateful. I think you have a future ahead of you. And I'm very glad to see the appearance here of what goes with this Francis Crick Award. And so I have pleasure in presenting Simon with the prize medal, first of all, and a certificate, and most importantly, a cheque.